Sir, good evening, sir. Sir, Jaganathan, sir. Jaganathan, sir. Sir. Audio is not, sir. Sir, audio, check the microphone, sir. Audio is not. Audio, hello? Jaganathan, sir.
Yes, sir. Sir, good evening, sir.
Yes. Good evening, sir. Now it is audible, sir.
Okay. Is now audible, sir? Am I?
Hello.
Hello, sir. Sir, am I audible?
Now audible, sir.
Yes,
You
sir.
are, sir? Okay.
I am Dr. Jagannathan, sir.
Yes, sir. Yes.
Okay, sir.
We'll start by 6:30, sir. Now 6:23 only.
Okay, no problem.
We'll start by 6:30. Okay.
Okay, sir. Okay, sir.
Yes, sir. Janardhan, sir.
Yes, sir. Yes, sir. I'm here, sir.
Yes, sir. I welcome all to the 13th day of Faculty Development Workshop on Pedagogy and Research Methods. Today's topic is vaccine conservation and production. Today, Chief Justice Dr. S. Jagannathan is Assistant Research Officer, Pasteur Institute of India, Nilgiris. He completed his PhD in microbiology in Baghdad University. He has filed 2 patents and he has published more than 30 articles in national and international journals. He has published 5 books and also editor in 5 books. He has arranged more than 10 conferences and is a member of the editorial board for many journals, both national and internationals. He has presented more than 20 papers in international conference and 5 papers in international symposiums. He is a member of 11 professional bodies. He has organized more than 20 faculty workshops and he has got more than 12 awards from various agencies. With these few words, I welcome you, sir, and I have this session to you, sir.
Yes, sir.
Thank
Thank you. Once again, good evening to all. First, thanks to Dr. Pungavanam and also my dear friend, Dr. Mani Jaiprakash, Register, Amat University. Today, I'm going to speak about conceptualization of vaccine. So here, I'm going to share my screen now. 1 minute. Sir, please allow to the screen sharing, sir.
Yes, sir. I will inform, sir. I will inform to them, sir.
Okay, sir. Okay.
System admin? System administrator? Karthik?
Share paniyachi, sir.
Okay, sir. That's I'm. Sharing. Okay. Now it's visible, sir.
Started.
Sir, now it's started.
Started, sir. Started screen, sir.
Okay, sir. I started that one.
Visible,
Yeah,
sir.
Okay, sir.
Okay.
Okay, so I'm going to that now. It is in off only is there. Now I'm going to fully increase that. Everyone now. Full screen. Okay. Yes, sir. Okay, now I am going to speak about vaccine conceptualization and production. Okay, I thought all are safe and all are enjoy the climate and they're in special things now. Today, the weekend of ourselves, we can start as a vaccine conceptualization and a production. In the first slide, you can see that 3 different figures. Number 1 is the great Louis Pasteur. He was the first person towards the development of vaccine. Towards the development of vaccine in the rabies, those was developed and first identified by Edward Jenner. So on behalf of Louis Pasteur, so our Pasteur Institute was started at 1907. So this is the entire our to rise, to yield, and so like Ulysses concept, that one. And also here you can see that Pasteur Institute building. It is a heritage building. It is more than 115 years old. Pasteur Institute. Actually, it was started for the vaccine production, especially for the rabies, towards the entire our nation. And also the structure of our Pasteur Institute, it will as a as a rabies virus structure. The rabies virus structure is a bullet shaped. Now I move to the first slide. Okay, what is meant by vaccine? It's a biological preparation. It can be administered through various routes, such as injection, inhalation, or oral, which stimulates immunity against an infectious agent. That is a biological preparations of vaccine. And also, usually, it contains a harmless variant of the pathogen. Pathogen means it is able to cause a pathogenesis. It leads to the disease. That means usually it contains harmless variant. That means it's not able to cause a pathogenicity. How it was going to develop? By killed or attenuated of the microorganism or laboratory generated altered of its toxins. That means the cellular parts or one of the surface marker. There's a spike and other kind of things. And next is that vaccine, it can be ability to a living being. For example, has, that means human being, animal, or any organism to resist and protect itself from the harmful effects of an offending microorganism or a toxin. Harmful effects against that thing. The efficiency of the immune system lies in its ability towards a recognize of foreign invaders without causing harms to the host immune cells and tissues. That means once you are vaccinated, that vaccine, it won't causing to our cells, also tissues. That means it can be recognized only to the foreign invaders. And also the vaccine also act by allowing immune system towards recognize and memorize the harmful viruses like bacteria and their toxins. That is a recognize the memories if it is coming to the later. And next is a booster doses. Booster doses means once you are going to vaccinate it on the 0th day, it will take minimum 4 days or 7 days or 12 days or going for a 15 days. It depends upon the vaccines or 1 year or 2 year. It's a booster dose. Booster of vaccine to boost the memory of immune system. That means already we are vaccinated on 0th day, so we have the sufficient amount of antibody. That antibody we want to enhance through the booster doses. That means enhancement by the increasing amount of the immunoglobulin G molecule and increase the ability to fight against the invading pathogen. That is the role of booster doses. Next, I move to the facts about the vaccine. The major contribution that vaccine have made, saving countless lives around the world. Every year, up to 4.5 million children's lives are saved by the immunization, as per the statistics of World Health Organization. And also, vaccination, it is a proven and one of the most cost-effective child survival interventions of the decades, 20 years, 200 years back. All countries in the world have an own immunization program towards deliver the selected vaccine to the targeted beneficiaries. I marked as a UIP. That means Universal Immunization Program. That Universal Immunization Program is especially focusing on our India. The world, entire world, it's a control by the World Health Organization. They could not interfere our vaccination strategy. So that's why each and every country having their own setup of their immunization practices. So one is UIP. Why it was started means each and every country it will be differ from the geographical distribution as well as epidemiological variation of that disease. So based on that kind of vaccines we are going to cover the entire nation. So based on that only our India we started the expanded program from immunization earlier. Now it's a converted and renamed as a universal immunization program to the particular targeted beneficiaries. Who are the targeted? Especially focusing on the pregnant women, infants, and children who are at a high risk of diseases preventable by the vaccines. There are today around 27 causative agents against which vaccines are available, and many more agents are targeted towards the development of the vaccines. But vaccines are not only for preventing infectious diseases, some help the body fight range of illness, activating the immune system. Yes, with those or immune system, it is now in the starvation period or it is not functioning. The vaccine is going to activate the immune system towards the recognizing as well as attack the disease as an invading system. The vaccine, what are the things it containing? It will containing as some of the ingredients. It contains some of the tiny fragments. That means a small fragment of the disease-causing organism or the blueprint of the making of tiny fragments. That means a disease-causing organism like some microbes. And also blueprint means there is either a protein, either it's a spike protein or it's a phosphoprotein or it's a DNA molecule or it's an RNA molecule. That is a antigen. And also it contains other ingredients. But what is the role of that? To keep the vaccine as in safer and effective, which vaccine component serves as a specific purpose. It can be intended for the specific only. And each ingredient is going tested during the manufacturing process. I will detail later. Okay. All the ingredients are tested for safety before going to the what I call it is an immunization practice. What are that? So number one is an antigen. Already I told it's an antigen. It's any kind of fragment of any disease-causing attenuated form, alive or killed form of materials. And next, stabilizer. Why we need a stabilizer? That vaccine, it is going for the various phases. Either it's a liquid phase or it is in a powder form phase. During the different type of condition, that vaccine should be as such kept in a as such condition. That means it could not be able to lose their specific activity. So regarding that, we can add the stabilizer, some of the protein like human albumin as a stabilizer, and also glycerol as a stabilizer, and glycine as a stabilizer. So different kind of stabilizer depends upon the particular type of vaccines. That is a stabilizer. And the surfactants. The surfactant, especially for the liquid-based vaccine. The role of surfactant is once the vial. So going for vaccine, going for filling in a filling vials. Sometimes that inner part of the vials they are underwent for washing as a sterilization and deinsterilization, steam solution and dry sterilization. Even though sometimes the inner part, the vaccine material is going to attach on the surfaces. So during the attachment also it leads to the formation of contamination. To avoid that, you can add it with added with contains surfactants. It should not interact with the surface of the inner part of the vials. Next, preservative. Here, the role of preservative means, so here preservative means to preserve the material to avoid the contamination. If the vaccine going for the long-term duration, especially for the liquid vaccine. That means if you are going to the hospital or any PhD or health center, so once the vaccine will going to open, so immediately it should be distributed. It should be going for vaccinated to the entire people. If they are going to give for something, that means though there is no patient available, there is no intended person available. So they're going to keep the particular time in the fridge condition. So it's already open. It leads to some contamination. How it can be going to happen? That means that fridge, it is not a sterile environment. It may be 0.0001%. So to avoid that, we are going to be using a preservative, like the thimerosal and a lot of preservative available. And the next is a adjuvant. Yes, the adjuvant is a vital role to the enhancement of the immunogenicity. Either it's a protein or it is a carbohydrate or any form of small glucose molecule, or the adjuvant, like chemical molecule like aluminum hydroxide or AM59 and various adjuvants available to enhancement of the antigenicity, that is called as an adjuvant. It can be along with that. And the next is a diluent. So diluent means it can dilute something. That it is going to help, especially for the freeze-dried vaccine. So generally, two kind of vaccine. One is a liquid vaccine, another one is a freeze-dried vaccine. Those freeze-dried vaccine, they're freeze-dried in a suitable condition. That freeze-dried vaccine only is going in stored under vacuum in a vial. Whenever we need, we want to go to that means diluted with a specific diluent, especially in that 54 millimolar sodium chloride, or it is in a water for injection. So that's going to dilute it. Then only it's going for proper dilution. Then only it's going for the vaccinated for immunization factors. So these are the main ingredients for the vaccine. That means antigen, stabilizer, surfactants, preservative, adjuvants, and diluent. Next, I'm moving to the next slide. Then types of vaccine. One is a LAV, live attenuated vaccine. The principle of LAV is it's a halted or the weakened form of the microorganism. That means a pathogen microorganism, which lacks of virulent, but it can able to retain the ability towards the creation of immunity against the particular diseases. That is called as LAV. For example, MMR vaccines against the measles, mumps, rubella. Then chickenpox vaccine, there is a varicella vaccine, and against is a oral polio vaccine. Here, I will share one thing, a role of oral polio vaccine. One thing, India got that. Another thing is polio-free status at 2014. And here, just 20 years back, India is having a lot of persons, a lot of children who are affected with the polio. So that's why the World Health Organization, UNICEF, and different combinations, what they're done, they want input as a mass vaccination campaign. There is a mass vaccination, which what we are now, we are moving for the COVID also. The same thing was happened for COVID. And every year in the month of January and February, those children the age of below the 5, they're going for vaccination. Because of that vaccination, it leads to that India's polio-free status. Last polio case was happened to India's February 14 at West Bengal in 2011. So that's why after the 3 years, so World Health Organization declared India as obtained the polio-free status, but still 2 countries have the poliomyelitis. One is Afghanistan, another one is Pakistan. If that country going to eliminate the disease, that entire world will get the eradication of the polio, and the one among the second to the next to the smallpox. So that is made up of live attenuated practices. Our next concept is a inactivated vaccine. That's a live organism are killed and inactivated and used as a vaccine component. They are usually heat stable and do not need an exact temperature. It need only over 4 degree, plus 4 degree, what we have in the fridge. For example, is a rabies vaccine, pertussis. So these are the inactivated vaccine. And the next is another type of vaccine is a toxoid-based vaccine. The toxoid vaccine, it is especially for the bacteria, like diphtheria and also like tetanus here. So some diphtheria, a Corynebacterium diphtheria, causing agent, and Clostridium tetani is a tetanus toxin. What we are doing in our PAS Institute, we are supplying the DPT group of vaccine to the entire country. So for the production of the Clostridium tetani, diphtheria toxin, we can, that is a vaccine strain. Like Clostridium tetani is a Harvard University strain. We apply to the Harvard University. They're going to cultivate in a fermentations, the suitable environment. So around 7 days in the process, that during the 5th days, that entire broth, what I'm speaking with is in 500 liter now. Once during the 5th day, that bacteria should not get much more of their nutrients. So it needs to the suicidal. So during the suicidal activity, that bacteria releases the toxin that is called as a tetanus toxin that is going to harvested. Then going for harvested, so immediately harvested, that is a toxin, then going for inactivation by the formaldehyde. Inactivation with the formaldehyde. So inactivation by formaldehyde, then that is called as a toxide. Then going for purification and the final formulation. That is a toxide-based vaccine. Tetanus toxide and a diphtheria toxide. And the next concept is here, newborn. That means called as a current knowledge we have. One is a subunit-based vaccine and also recombinant-based vaccine and a polysaccharide and a conjugate-based vaccine. These are produced by employing more modern technologies by purifying and isolating the active components of the microorganism which produce immunity but not disease. For example, say hepatitis B vaccine and HPV vaccine. Now I'm moving to how the vaccines are developed. So here, what I detailed that one is what is vaccine, what are the ingredients, what are the plus point of vaccines, what are the different types of vaccine. Now I'm moving to how the vaccines are developed. Okay, so here there's 2 different types of technology there. One is variolation method, what I call it BCG vaccines, variolations, and also smallpox variolation vaccine. And a killed organism, like the polio or and other kind of this, killed the organism. And it's a live attenuator. Earlier I told is an OPV and MMR like that. Okay, and next a polysaccharide vaccine. Polysaccharide means it is a pneumococcal-based vaccine, and the next is a cell culture-based vaccine. The cell culture-based vaccine for the cultivation of any kind of virus, we need the virus cells. It need a live cells. That is a tissue culture method. So these are comes in the category of empirical knowledge. That means what we gained based on the tradition we follow. That's the empirical based. And the next is a modern vaccine technique. One is a glycoconjugate vaccine. Glycoconjugate means the surface-based protein. There's surface-based glucoses that is going for conjugate. That means conjugation of various type of serotypes. Like for example, it is in a Haemophilus influenza. It's containing 13 serotypes there. From that, each and every serotypes, that conjugation of that kind of carbohydrate, they're going for, that is called as a serotype vaccine there. Then recombinant DNA, classic hepatitis B. And the next is a reverse vaccinology. Reverse vaccinology means previously what we are doing, we are going to identify the pathogen, that pathogen going for the screening, then going for sequencing, then going for find out the entire thing. But currently, so once we are find out the disease, they immediately going for sequence the entire thing. So after the sequencing, then going for find out the antigenicity of that the neutralizing antibody is present, using with the bioinformatics tools. And the next is structural vaccinology. That means what kind of structure involved? Either it's an outer membrane protein or inner core protein or a nuclear core protein or it is a spike protein. So each and every structure, that means contained with the microbes, that structure going to useful as a vaccine system. And also adjuvants like ISCOM and also ISCON and the liposome and different kind of adjuvants available for the releasing of vaccine towards a targeted delivery. That means going for intramuscular or intraperitoneal or intraperitoneal or subcutaneous injection. So we are just piercing the skin here, but here this kind of adjuvants, we can go through the liposome. The liposome means is a targeted delivery and is a nanoparticle. So nanoparticle based vaccine is emulsion based. Either is an oil in water. That's it. So these are the vaccine comes under the technique. It is called as modern vaccine techniques. What is it? Like a conjugate, recombinant DNA, reverse vaccinology, and the structural vaccinology, adjuvant, and the liposomes, as well as nanoparticles. And the next, okay, here I have something that is called as a vaccine development. You can see this thing, what you call, this is a slide, this kind of needle-based structure. It results year of the infection agents. Year of the infection agents was linked to the disease, and here the vaccination was licensed in the entire US year. First starting with the 1938, we identified one disease that is called as influenza. What we are facing, same kind of things happened in the first pandemic before the plague. So 1938, we identified the influenza, and 2004, we got a very good vaccine. Just point to this, very good vaccine means a highly potency, highly efficacy, and also highly safety and highly immunogenicity on the 2004. Then only it was approved by the US FDA and immediately go to the entire world. Next is smallpox. But where it was started, where it was ended, we does not know. But that was concept, the great man, Edward Jenner, we can salute him. That Edward Jenner, he only a person was that variola-based vaccine. He was find out the same type of concept. It was further developed and further characterized in 1949. Finally, we got the smallpox vaccine. In malaria, we identified it in 1880. Still, there is no proper vaccine available. Still, that entire research are going on the entire world. That means, but some of the drug available, anti-malaria drug available, that drug, it can be effective in the particular person. So that's still there is no vaccine available. And the next is tuberculosis. We identified in the 1882. From 1882, that also is not having the proper vaccine. Only we have the BCG, which is like Almantagurian vaccine, what we are giving to the newborn baby of child and below the within the day of 3. That also has some particular control, not the entire thing. And the next is in 1884, identify the one fever that is called a typhoid fever. So the vaccine we find out at 1989. Like the meningitis, 1889 we got this one that they identify the meningitis. In 1981, the vaccine was added to the US immunization schedule that the country is the entire world. Next is a pertussis, that is a whooping cough. 1906, when the passenger was started for construction. So that time, the pertussis whooping cough disease was identified in 1906. In 1948, immediately the whole cell pertussis vaccine was developed. And dengue fever in 1907, the identified still, we are not having the very good vaccine against this thing because of the 4 different serotypes. 4 different serotypes, that strain variants. So all having the various kind of strains. For example, why it is a problem has happened in dengue cases. Here, if a person, if a vaccine was started the strain A, if going to vaccinate a person, that vaccine belongs to strain A. But that person is going to bitten with that mosquito. That mosquito having this strain B type things. So strain B type of that virus, dengue virus. So that antibody, it cannot be invaded, it cannot be neutralized. That thing. So that is a failure of that things. So that's why the dengue vaccine in US, they are now combined with all 4 serotypes. Now it's in that kind of phase 3 activities going on, still not in successful things. But the vaccine now they are treated to the military people of US now, not successfully come to the publics. And 1908, the polio we eradicated in the world at 1955. And this is not curable, but vaccinates are able. So previously, a person was vaccinated by oral polio. Now they are injected to the inject-based polio vaccine is here. That also developed. And the next is a Zika virus. Is a 1947 is coming. It came on 1947. 3 years back also in India, we faced one thing, Zika virus, our neighboring state of Kerala. Still, there is no vaccine available. That next is chicken pox. In 1953, we identified, and on 1995, we got a vaccine against that chicken pox. That variant vaccine. In measles, we identified on 1953. That also having the vaccine in measles, mumps, rubella. And like 1960, we identified a disease called mononucleosis, and hepatitis B. 1965, we identified. At 1981, immediately that is a first vaccine. So identified 1965. So immediately we got a vaccine at 1980 within a short period in the entire history. That also take from 65 to there is a 14 years. That is a very very less time period taken for the particular vaccine. It's a successful vaccine. That is a hepatitis B. And next is a rotavirus. We identified 1973 and also vaccine against 2006. And like Ebola. Ebola vaccine was released on last year. There's a 2020 in that US-based company. Now is in it was in the final trial and released that thing. And the next is a cervical human papilloma virus, a cervical cancer vaccine. And these are the things is a vaccine development. And the coming slide, I got details about we can towards the vaccine development. So 3 Ds is important. What are the Ds? One is a discovery. Discovery means what? We can discover one thing in the lab conditions. Then we moving to the process development, research development. Here, process development means to produce an economically viable vaccine, consistent in a manner that satisfies the regulators. Just to find out what is meant by regulators. Regulator means those person. That means it's a national regulatory authority. In India, we have that. We are following the Indian pharmacopoeia. In Japan, they are going to follow it in a Japan pharmacopoeia. In US, they are going to the US pharmacopoeia. In Chinese, they are going to follow the Chinese pharmacopoeia. So each and every country having their own, it is an own mandatory document. So that mandatory, what is a mandatory test parameters? So what vaccine we have, that vaccine it should meet the regulatory norms. So in India, we have the Central Drug Laboratory, Central Research Laboratory. So they are our things. So during the process development, there is a pilot scale level. We are going to the making a bulk. So after the bulk, the manufacturing, we are going to product finishing. So once the bulk was started, they're going for product finishing with a fill and finish with the vials and other things. That's processing. Next, I'm moving to the clinical development. So here, clinical development only is a phase 1, phase 2, phase 3 manner. So here, they demonstrate that the safety and measure the protective effect of the vaccine in humans. So these are the 3 Ds. And the final Ds is assay development. Why the assay development? Once the person going for vaccinator. So that vaccinator, we want to check the vaccine, whether it's can be able to produce antibodies in the person. There's a neutralizing antibody. that was started old. So these are the things we can we should have to keep the mind of to check the vaccine. This thing. So the entire thing we may develop in the process for the assay development. So 3Ds. One is a discovery. Once the discovery started, then the PD is a process development, and the CD is a clinical development, and the AD. So start with the PD and come back to the AD assay development. So these are the 3Ds going for towards the vaccine development. I move to the next slide. You can get a very clear what I explained in the previous slide. Yes, PCT. That means preclinical research. Here we are moving to the one thing that is it's called as a establish vaccine candidates as desired effects in a in vitro. It means in a laboratory condition. So once the laboratory condition means like is a chemical analysis and the pH of the vaccine, and also the TDS of the vaccine, and also protein content of the vaccine, and everything we are clearly entered in this thing. And next, moving to the effects produced and consistently. That is in preclinical. Once we reach the successful results, so immediately we are moving to the next state. Is called as animal studies. That animal studies, it is a desired human response. It protects animal from the disease. That is, it is called as a what do you call it? It is a in vivo, in animal-based that. For the in vivo, it protects animal from the disease, and also it can ensure the relatability of the free offsite effect. Then once we received the successful results in that animal studies, that means in vivo, now moving to the human trial. It leads to the three type of trials, and the final is the fourth side. One is the phase 1, phase 2, phase 3. The phase 1, it leads to the less than 100 healthy volunteers. Just please understand it. Please understand this is the healthy volunteers. And also that we choose the less than 100 volunteers. Maybe those who are working in the laboratory or those in the production area, they are combined, started in the particular production area. Next, during that phase 1 studies, they are moving to the escalating the doses into this. Escalating means which like 0.5 ml, and then 1 ml or 1.5 ml or 2 ml. That's the escalating doses, depends upon the health. In this phase 1 trial, same kind of gender people, and also same age group, and also same weight of the peoples without the comorbidity peoples. That people were taken and going for the vaccinated. One person going to 0.1 ml, then going for 0.2 ml, and 0.5 ml, then going for 1 ml. So based on that, we are going to check the efficacy of the vaccine. That means safety as using the blood sample. That means the entire biochemical marker and other physiological marker, everything you have checked in a phase 1. Once it succeeded, then moving to the phase 2. Here we are moving to the less than 1,000 and also moving to the 100% volunteers based on the intended and the age for disease. For example, this vaccine moving for the children means we are going to intend for the children. If you're moving for the kids, we are moving to the kids. If you're moving with the adult, it's going for the adult. And also during this phase 2, we are moving to the wait for the additional safety. That means safety means that vaccine it should not giving the trouble. Trouble means either a death or any kind of things, and also efficacy testing. Efficacy means the immunogenicity. How much volume of vaccine they are vaccinated, then how much are we expected the antibody, whether it's released or it is not released. That thing we carried out in a phase 2 trial, especially for the immunogenicity, already at the immune response. Here, immune response means they are going for vaccinated. So we have the immune cells, either T cells or B cells. So that entire cells, if you are going to the properly secretion of the antibodies, either the IgG or IgM or what kind of antibodies, whether the IgG, the particular antibody, it's a specific for what we are vaccinated. Whether otherwise a non-specific of that thing. So the entire immunological characterization need to be done in a phase 2. Next, moving to the phase 3. In the phase 3, it's 100K volunteers, and also it's going for expand the safety database. Either say phase 1, phase 2, phase 3. Every time we are consider about the safety of the things. That means establish a protective efficacy as well as clinical consistency. Then going for market release, and further moving to the phase 4. There's a post-marketing surveillance. Yes, once the vaccine was released, then going the post-marketing surveillance. So that's why each and every person, those who are vaccinated, their entire detail history, everything collected to check the adverse effects. To check the adverse effect means some may be. So those type of results we are not ascertained during the phase 1, phase 2, phase 3. And also those results we will not face during the phase 1, phase 2. It may be happen. It depends upon the personal things. So the entire thing is going for, this is for assess the long-term efficacy. So phase 4 only succeeded, the immediately the vaccine going for the releasing. So these are the various phases in a vaccine trials. One is PCT in vitro animal studies, as a in vivo, as a phase 1 and a phase 2, and final phase 3 is a phase 4. These are various phases. Now I'm moving to the next slides. Okay, these are the things. Okay, why the vaccine development? I'm looking at some of the failure. Yes, less than 1/10 of the vaccine candidate to achieve the license. Just imagine, 10 companies going to start the vaccine. But out of 10, only one vaccine is going for licensure, what I explained in the previous slide. So on the next also, the high failure rate is due to the unpredictability. That's the biological unpredictability. If I'm going to vaccinate today one person, this is a same age, but that vaccine it giving the higher amount of immunogenicity, higher amount of immune response. The same vaccine I'm going to give to the same age group, same age and the same body weight and everything, but it is not release a much more immune response. That is a higher failure rate in the vaccine. There is the unpredictability of the biological organism needed to produce a vaccine. Due to the uncertainty, how the human, our immune system will process and react the vaccine antigen. Depends upon the immune system. So we will not expect that person will get about 2.5 IU international unit, or this person will get 1.5 U. So it depends upon the personal immune system. How the person is going, how the immune system will get trigger. After the how it is going for activator. Then after activation, how it is going for enhancing. Then finally, how to enhance of the antibody, and also that enhanced antibody, then going for how it is survived, and also how it's going for longevity. So these are a lot of the factors involved in it, this immune system. That's why it is a vaccine development is a higher failure, right? And also some vaccine candidates, it may be produce appropriate levels of immune response. It's good, but it induce adverse events, reaction. That means it's going to failure. But it's 100% in the appropriate levels immune system. If leads to the failure in the person, adverse reaction means it's a failure. It's also failure. Like other vaccine candidate, it may be safe, but it's ineffective. There is no effective, and efficacy should be less than. So these are the failure of the vaccine development. That means less than 1/10 of vaccine candidates going to licensure, and higher failure rate because of the unpredictability, and also higher amount of immune response if it having that adverse reaction getting down, and other vaccine candidate may be safe and ineffective. So these are the things for the failure. And further, okay, why the failure of the failure to produce a vaccine? That is the evasion mechanism. All microbes deploy the evasion mechanism that interfere effective immune response. For many microbes, it is not clear that which immune response provides the effective production. Still, we are not having the personalized medicine. Individual person, we are not able to identify how how the immune system going to functioning. And next is a limited range. A vaccine given tends to be an effective only against a particular individual, say serovirus of pathogen species, although some species have 100 of serovirus. Okay, that means we are going to develop a vaccine. What during that first time it will having the only particular specific strain. Later, after development of vaccine, it may be the different type of serotypes. So that's why that vaccine it is not be act on that things. But some of the few percentage, only some few 60 to 70% of the conserved genes will be there. Other things going for the happen. There is a point mutation or anything going to happen. So that's the strain variant. So if you are concentrated focusing on the particular vaccine, it can be for antibody that little bit only. We could not expect from that is it depends upon the serovirus. That means the strain, substrain. And also, disease is not immunizing. For some pathogens, even exposure to disease does not confer active immunity. And the next is a rapid evolution. Development of vaccine against a particularly rapidly evolving pathogens because of the RNA. What RNA is going for reverse transcriptase. So reverse transcriptase, if you are going to develop a vaccine, so immediately that is going to not properly invading system, properly neutralize that things because that viruses, it is what virus is having, is going for multiplied based on the reverse transcription. It's also difficult. So that is a problem for the, there is no development of proper, there is no vaccine against that things. And next is the exacerbation of the diseases. Vaccines on certain types against certain pathogens can actually exacerbate against a disease when it is occurs. So these are the failure to produce a vaccine. And finally, the cause of the disease. And live vaccine retain at least some potential for causing the disease. This is especially true to regard the immunodepressed individual. For example, live polio vaccine. Once a person going for vaccine with live polio, there is a live polio, there's a vaccine reverted disease also happen. That is leads to the cause of the disease. So these are the things for the failure mode. And finally, is a cost-benefit problem. Successful vaccine delivery, it is not always economically justifiable. So it's a failure to produce a vaccine. There is a vaccine development things. Okay. So now you clear that how the vaccine was developed and why the vaccine failure was happened, what are the things were there. Now moving to the, what are the techniques in vaccine development? What is the gene technique is there? So techniques, vaccines are currently produced by the gene technique, and instead of using virus or bacterium things, a single gene as history surface of glycoprotein of the viruses, it can be expressed in a foreign host by cloning. So all knows that either using with expression vectors are used to make a large amount of antigen to be used as a vaccine. Most used vector for expressions are either a bacteria like E. coli or the baculovirus system or yeast, yolk, anything. And next is this process induce the vector to produce an antigen, which was then purified for further activity. The purified antigen when combined with an adjuvant results of safe and effective vaccine. So this is a gene technique. And for example, as a human papilloma virus vaccine, that is very effective preventing of the cervical cancer by developed with the BLP vaccine-like particle. And the current EBI will also develop the gene technique method. So during the vaccine development, this technique called as 3 T's. One is a transduction, transfection, or transformation. So transduction means it's a foreign DNA, whatever desired is, introduced in a cell by a viral vector. For example, it's a bacteriophage infecting the bacterial cells. It is also described the introduction of a recombinant viral vector towards a target cell. That is a transduction. That is a transfection, and the infection through the transfer. That is a post introduction of the small molecules, such as a DNA and RNA directly to the eukaryotic cells using with the electroporation, or calcium phosphate, or gene gun, or a micro-injection based. These are the things are used for the infection. And the next is a transformation based on the Avery McCord. You know that. The DNA uptake by the bacterial cells from the environment. That recombinant plasmid DNA can also be introduced into the competent bacterial cells that take of extracellular DNA from the environment. So 3 T's are important for the specific for recombinant DNA vaccines, or it's in RNA vaccines or mRNA vaccines, or going for DNA-based vaccines. So entire thing, the 3 is a main concept: transduction, transfection, or transformation. And next, how the vaccines was developed using the genetic engineering technique. So selection of the gene of interest. So immediately that same times we are going to select the plasmid. So plasmid either is a cloning plasmid or going for the expression vector. So once plasmid selection, then going for the vector. Plasmid selection going for a vector. We are going for the suitable method. Here, selection of interest either is a bacterial or gene or protein anything. So once it's selected, we may go in for the based on the science. and based on the DNA manipulation and which expression vector we are going to use that kind of factor that. And next is here selection of vectors. Either is a pouch or cosmate or backlow artificial chromosome or yeast artificial chromosome expression vector. And next moving to the selection method, either going for direct inoculation to the eukaryotic cells or going to using with the expressed protein. And next is selection of the technique, either micro projected by the bombardment or going for electroporations or microinjection or recombinant DNA. So these are the technique involved in the vaccine development. For example, it is some flowchart diagram. That flowchart means first a selection of the gene of interest, then selection of the plasmid, then a selection of the vector, and the selection of the method, and the selection of the techniques. So these are about this thing. I'm moving to the next slide. And also next is a viral antigen generation, all of the gene cloning technique. Here, the gene for the viral antigen of interest, what we need now, okay, it's cloned into the another organism in case of bacterium and exposed and produced in a very large quantity, like hepatitis B and diphtheria, cholera, and tetanus, some program. So it is a virus particle. It's a virus particle. So we are moving to the nucleic acid. That is the antigenic protein. So from the DNA transcript, we are using that. So if a gene was unwanted gene was excised, then going for needed gene going for inserted in the desired plasmid, then going for expression vector, the bacteria producing antigen that is using with that pure antigen moving as a vaccine. So here, and another thing is a surface antigen. The plasmid with the GNA into a surface antigen of what it decided as any viral component. The surface antigen is released to that what is a yeast cloning vector with a synthesis and release the surface antigen. The synthesis product only we are going to be using as a vaccine component. So this is a viral antigen generation. And also how to construct a recombinant attenuated viruses. Attenuated, yes, which first we isolate the pathogenic viruses. That is the isolation of the pathogenic viruses. Then moving to the once is meet that entire criteria what we have now. So then moving to the cloning of the genome. Either it is in a suitable vector and a suitable condition. The condition means any kind of experiment that material what we are going to the cloning, what we are going to the need a virus and a protein, anything, that protein, it should not underwent for the denaturation or gene underwent for the any kind of losing the specific activity. So that's why the condition is more important, so cloning and other kind of things. Next, the isolation of virulent genes. By virulent genes with a suitable assay test and going for mutate and delete of the unwanted material. So after the mutation, that is called as a viable, the result of viable and immunogenic avirulent vaccine candidate. You can get much more clear idea about with the picture. In this picture, so here the isolated pathogenic viruses, then going for clone the entire genome. Once the cloning was happen, that is this is a blue color. It's a receptor binding protein gene. So here this red color is a virulent gene. The green is a capsid protein. So entire thing we are excise that. So then going for we are moving to the isolated virulent gene. So that we mutated, there's a mutated virulent gene, and also delete the virulent gene. So that mutated virulent gene resulting virus is visible and immunogenic but not virulent. This may be used as a vaccine. So mutated virulent gene vaccine or deleted virulent vaccine. So these are the construction of recombinant attenuated viruses. One is isolate the pathogenic viruses and going for cloning with a suitable vector and a condition, suitable vector condition and a suitable assay test, as was the deletion of the unwanted and the mutated as a virulent gene, then is moving to the viable immunogenic and the virulent vaccine candidate. Next, I'm moving to the next slide. Under subunit based vaccine. The subunit based vaccine, it's a one that only uses a very specific parts, like something spike, like something in this phospho, like inside the small spindle, like the of the virus or bacterium that mimic that immune system needs to recognize. And also another thing, how this gene or this protein we are going to select that based on the neutralizing antibody capable. That means where it is going to bind our body whether it's capable of neutralizing antibody synthesis. So that kind of part only we are going to concentrate for this kind of vaccine. And also it does not contain the whole microbe. It's a subunit. The name is a subunit microbe, or use a safe viruses as a vector. The subunits, it may be in proteins. Sometimes there's in a that kind of, what do you call it? It is in a protein of overall coverage protein, or it is in core protein. Sometimes the sugar molecule. That means the glucose also there. Carbohydrate based. And most of the vaccines on the childhood children are subunit vaccine, protecting people from the disease such as whooping cough, tetanus, diphtheria, and meningococcal meningitis. So you can get some figure here. Only uses the very specific parts, that is subunits of a viral or bacterium that the immune system needs to recognize. That is a recombinant DNA vaccine like subunit. And why further details? Here, recommended very subunit, like clear that isolation of the viruses, final mode, the extraction of DNA, and the isolation of desired gene of interest. Next, I'm going to using with one thing that is called as a expression thing. There's a non-pathogenic expression vector. The vaccine of viruses are going to isolate the vaccine DNA extraction and recombinant restriction to the digestion. That means cleaving. The ligation of the RE digested vaccine virus. So that surface has a desired gene. I'm going to recombinant. That means recombinant with the non-pathogenic vaccine virus system. That is going for transform its suitable host, then going for upstream process, then downstream process, fill finish, meet the regulatory norms, then vaccine. That having bigger, yes. So the DNA, for example, herpes simplex viruses. That herpes simplex viruses, I'm going to develop a vaccine again, HSV. So that herpes simplex viruses, DNA is going to extract that extracted DNA. I'm going for the as a polydigestion, recombinant, extraction to digest. Then gene was isolated, the gene specific herpes simplex protein. So here also we can get that outcome of the results from that reverse vaccinology or bioinformatic tools. Then that gene, and simultaneously I'm going to the harmless vaccina. That's a Copox viruses. It is harmless. That vaccina DNA as a sample is extracted and cleaved with what enzyme I used in that RE, the same for here. Then going for fragment content gene and going for cloning here. So that the harmless engineered viruses, that vaccine with surface like herpes simplex injected in the human body. Unreleased things. So this is called the recombinant DNA vaccine, which is the subunit of the expression vector. Is a harmless vaccine virus. This thing, and then moving to the nucleic acid vaccine. A nucleic acid vaccine just uses a section of genetic material that provides the instructions for specific proteins, not the whole microbes. So DNA is a first turned into the mRNA, which is then used as a blueprint to the make it specific proteins. A nucleic acid vaccine, it can able to deliver a specific set of instructions to our cells, either as mRNA or DNA for them to make the specific protein that we want our immune system towards a recognize and respond it. The DNA vaccines are the vaccine which contain DNA that codes express the specific proteins from a pathogen. That is a nucleic acid vaccine. And also the DNA is injected into the cells, uses the DNA, synthesize the protein. Okay, that's the translation process. Because these protein recognized as a foreign, when they are processed by the host cell and displays as surfaces, the entire immune system is altered, which then trigger the immune response. Those are nucleic acid vaccine. So normal vaccine provides primarily humoral immunity, but on the other hand, DNA vaccine both humoral as well as cell-mediated immunity. Once again, I detail you. So nucleic acid vaccine just uses a section of genetic material. It can able to provide the instruction-specific protein. It's not whole microbe. First, it's the DNA converted to a mRNA. Then DNA vaccine are the vaccine which contain DNA that code a specific part of the protein. For example, particular protein, the decoding of the DNA, that DNA only directly inject to the cells. The immediately is going for surface altered that After the altered immune system, it's a trigger the immune response. So generally, the traditional vaccine it going to cause only the humoral-based immunity, but the DNA-based vaccine it can able to cause based cell-mediated as well as humoral immune response. So here's a genetic material approach is a vaccine uses the genetic material for specific proteins is a DNA is or RNA. So this kind of things. And the next is a recombinant hepatitis B vaccine. How it is going for develop or produce there. So here hepatitis B viruses. So the DNA or anything, or the hepatitis B antigen continue. There's a recombinant DNA. Simultaneously, we are using the bacterial DNA. Then going for plasmid DNA, it's going for cut without expression enzyme. But recombinant vector further going for expression system. Why? Okay, you have the doubt. Directly you can move with this kind of bacterial means. So here why you are using the recombinant yeast cell? That means yeast cell means it can be grow easily, or and also it cannot easily contaminated, and it may be in it releasing the surface proteins the more. So that's why this kind of expression vectors are used in a production activity, the fermentation, actually the antigen. Then finally, once the fermentation was completed, the recombinant yeast cell multiplying and producing hepatitis B antigen in a fermentation tank, and finally going for extraction. That means the DSP, downstream processing, purification, as well as the hepatitis vaccine. Next, finally, is a HB vaccine. Next, the DNA vaccine is a schematic representation. So the entire viruses, it contains RNA and the virus cross sections and envelope protein and E protein or M protein. E protein is envelope protein, M is membrane protein and pre-membrane protein and a lipid bilayer entire things here. So we are going to cut with the pre-mem protein here. Once a cut, then we for the RT, reverse transcription by the RNA to DNA mechanism. Then DNA going for the entire sequencing where it is located. What is the entire sequence synthesizer? So DNA onto the DNA sequences, they're further going for sequences here. Once the sequence is based on the DNA sequences, primer short sequences of the DNA specific of the pre-M and these primers are which they turn into the used to the generated cDNA fragment. This is cDNA fragment, so containing both the pre-membrane protein and these genes. That is going for a clone in it. Our desired things that after cloning, so carrying the agent, further going for the downstream processing is a column chromatography and clarification of the 0.2 to 0.45, then purification. That purification is called as a vaccine. So this is a schematic representation of the how the DNA-based vaccines are produced in the world now. And the next, once the vaccine was produced, then we are moving to the DNA vaccination. So DNA vaccination is a technique for protecting an organism against a disease by injecting it with the genetically engineered DNA towards a produce an immunological response. Circular double-stranded DNA molecule, it can be referred to as a plasmid fabricated with a DNA sequence containing genes encoding one or more proteins of the pathogen. At this DNA inserted into the cells, it is further going for translation product, translate to form as an antigenic protein. As a protein foreign to cells, so the immune response rises against this protein. That's a DNA vaccination. And how the RNA vaccine are going to start that? It's a target protein, a target pathogen that going for genome sequencing. So once the genome sequence is done in one particular country, then immediately the transfer of the sequence to all the world, wherever the interlinked connected. Then going for vaccine design using that reverse vaccinology. So the vaccine design then going for the mRNA production purification. After this, formulating testing, the pilot mRNA vaccine production, then going for GMP. That means good manufacturing practices, mRNA vaccine, then vaccination. There's a RNA-based vaccines, what we have now also. Then viral vector vaccine. And this type of vaccine uses safe viruses towards the delivery of specific, to the delivery of specific substance called the protein of the germ of interest, so that it can trigger an immune response without causing disease. To do this, the instructions for making particular part of the pathogen of interest are inserted in the safe viruses. The safe viruses then serve as a platform or vector to deliver the protein of the body. For example, it's an Ebola vaccine. The trigger protein only development of these things. bigger protein is part of this thing. And the next is the Ebola vaccine is a viral vector vaccine, and this type can be developed rapidly. There is a viral vector vaccine. Here, the two type of viral vectors. One is such weakened viral vector and replicating viral vector and non-replicating. Replicating is example is a measles-based things, and non-replicating is adenoviruses based. Okay. Here, such the newly approved Ebola vaccines in the world, what I told earlier, that replicates within the cells, such vaccine tend to be the safe and provoke the strong immune response. Existing immunity to the vector could blunt the vaccine's effective. However, the coronavirus spike gene and the virus gene is a viral replication, like is a adenovirus based. The Johnson also now doing the vaccine again, this thing for corona spike protein. And also now in type phase 3 trial, there is a viral vector, also replicating and a non-replicating based that. Next is another concept about VLP vaccine. What is a VLP? Virus-like particle vaccine. Once we isolated the viruses there, so immediately we could not able to handle the viruses in lab conditions. So we need a BSL 415 conditions. So here, that virus, some of the particles, there's a virus-like particle, there's a virion. That virion is like a pseudovirus, and also it's called the pseudoviruses. Here, it could not be able to cause a pathogenicity, but it can be able to elicit immune response. That's the virions. That virions further going for the polymerase chain reactions for the amplifications. Why we need amplification? During the amplification, what our desired virions is going for amplification, that we got some things, amplified fragment there, that amplified fragment, we're going to again ligate it to the expression system. That expression system then going for expression in eukaryotic cells, either is a yeast or any insect cells. So that insect cells can be able to produce something that is called as a VLP, virus-like particle, that protein, expressive protein, what we are cloned in expression vectors, what we are amplified that is. So that means the purified VLPs, again we are going to add it with the adjuvant, so that adjuvant, it can be enhanced with things that is called as the vaccine, then going for clinical trials. There is a VLP, virus-like particle-based vaccine. So here, in currently, one vaccine that is successfully under the immunization program, that is called as a human papilloma viruses. Again, the human papilloma viruses, especially it is used to the girl child for the to prevent cervical cancer. That's a VLP-based vaccine. Okay, I thought you may be clear about how the vaccines are development, what are the techniques involved in vaccine productions and development. Now we are moving to the vaccine production. Okay. Vaccine production here. Okay, so now I am from the past history of India. Why it was started? It was started for the rabies vaccine production. So, rabies vaccine production, along with that, we are in the production of DPT group of vaccine, DPT component, pertussis component, tetanus component. So that the entire DPT group of vaccines are produced for the newborn baby and children for the immunization. The tetanus toxoid vaccine we are going to using for the pregnant mother as well as the septic injection. Apart from that, we are in the production of rabies vaccine. From 1907 to till date, we are still producing the vaccine here. Okay, what is rabies? Rabies is a zoonotic disease. So the disease that can be transmitted from animal to humans, especially it can cause by the rabies viruses. It's a lexavirus genus within the family of Rhabdoviridae. That virus, it can be easily transmitted in the saliva of the rabid animals. Generally enters the body via infiltration. That means a virus will add from the saliva of rabid animal into the wound, either with the scratches or the direct exposure of the mucosal surfaces to the saliva from an infected animal. For example, it's a bite. And also, please note that it's 100% fatality happen if a person having bitten with dog, if we are not properly treated. But it can be preventable. It can be preventable if you are going for the proper medications, the proper protocol, if you're going to follow. Most commonly, It's a 95%. It's a 95%. It is a transmitted from the dogs. Sometimes less than 4% by the monkeys, donkeys, horse, cows, buffalo, goats, sheep, and pigs. Sometimes, occasionally, is a wild and mongols, fox, jackals, camels, and elephants. This is a 100% of this thing. So moving to that. Okay. In this slide, I'm going to show that out of the furious and paralytic rabbits and rabbit raccoons, you can see that how it is a gentle. It's doing that. It's this. You can see that. Look that. It can some of the drooling of saliva is coming here, and also it being a highly furious form is going to tend to bite to others here. The next is here. Salivation, it is beyond that screen. It's going for coming, and also the red colored tongue inside that is a white colored saliva. It's coming out. It's a drooling of saliva. And the next is here the raccoon, paralytic rabbit. So it's a rabbit raccoon. So you can see that it is containing that sticky in that tongue. That is also in saliva contained rabies. Under the great another photo, you can see the dog, it containing something that is a saliva. It is a what you call froth-like nature. It's like froth-like, and it's going to tend to bite others. And these are things. And another thing is the great rabbit raccoon. See that how is furiously is there. In that condition, if it's going to bite the person, it's immediately going to reach the brain. Okay. In this slide, I will cover the things. How the rabies virus are transmission. You can see the dog. I thought those who are attending here, they knows that they're having a lot of pet dogs there. So I'll get a small, what I call some tips about how to check is a normal dog or it is a rabbit dog. I can look there. Dogs are based on the color of the dog saliva. If dog saliva, if it is in white color or is less watery, means less watery and less viscosity means there is no virus particle present. There is no rabies. There is no chances of rabies virus particle present there. The same saliva just little bit change the color into the yellow color. If there's a pale yellow, that means that saliva containing 50% of the virus particle. Pale yellow color. Then our next stage is if it is in a brownish color. That means a pale brownish color. So if the pale brownish colors happen, so the 100% of the saliva containing the rabies viruses. Okay, white color, non-sticky and less viscosity, and also is watery means there is no chances of virus particle present there. And the next is yellow color means 50% virus particle. Next is a pale brown color means 100% virus particle present. And the next is then what is a rabies particle content? It's a rabies virus. It's a bullet-shaped viruses. I shown in my first slide why our passage was started. 5 is a construction of 4 different based on the structure of this thing. Bullet-shaped that. So rabies is a bullet-shaped and also it's having the different kind of structural and non-structural protein. Like G protein, L protein, G protein, L protein, M protein, premembrane protein, and a nucleopore protein, and genetic materials, the single-strand RNA viruses. So that is present in a dog saliva, either it is in a yellow or brownish color. Then the dog going to bite a person on that this thing, virus inoculated. So once the virus was inoculated, immediately where it is moving, then moving to the virus replication in the muscles. So once going for replication by like this kind of things, and moving to the virus bind to the nicotinic acetylcholine receptor. That is a neurotransmitter. It also going to the neurotransmitter junction present there. Once it's bind that, so immediately the virus travels within axons of the peripheral nervous system via a retrograde fast axonal transport by the dorsal root of ganglia. So once it's reached there, the replication of the motor neurons of the spinal cord and the local dorsal root ganglia in the rapid ascent to the brain. Once after reaching there, the infection of the brain neurons with the neural disinfection. Then finally, the entire centrifugal spread along the nerve cell, salivary glands, skins, the cornea, and others are organs going to the failure. Okay? Once going to the bite area, then immediately on the virus replicates the muscles. So immediately on that is going to bind to the neurotransmitter. That once the neurotransmitter acetylcholine dopamine, it's going to, based on that, it's going to reach to the dorsal root ganglion, to reach to the spinal cord, or to the brain, finally reach to the brain system. Okay, once it's reached to the brain, what will happen? Yes, virus, it cannot infiltrate into the skin. But once virus reaches the brain, it further replicates and resulting and present clinical signs from the patients. What type of signs? Here, you can see that how it was happened. Okay. Here, why this why the persons was behave like a dog. If a person was bitten with dog, why the center behave with a dog? Because so once it's going to that virus reach to the brain, the main function of brain is to control the entire system for the visual, for the hearing, for the control the striated muscle, entire thing. If brain is not giving a proper signal to the eye, so eye also you have the cone and rod cells, you cannot be able to visualize anything. That is called as a photophobia, like both ears. And also here we have that what is called here nasopharyngeal is having the pharynx and pharynx region. That pharynx and pharynx region, there is a chances to that it is moving for that what is the transport esophagus. That is the esophagus is a main purpose to that esophagus. The main purpose to the what do you call it? It is going for food transport. So that also the signal from that side, there is a metaphor placement signal from the brain cells. If it is not properly transferred, what happened? Because of fully contracted. It's fully contracted, there is no proper releasing means. So that means it leads to the not allow the food inside there because it's a contracted. And also the sound. What we are going to speaking, that sound organ is not perfectly coming out. If you want to make a sound, that may sound look like as a itching, like a dog sound like that. Like finally, that is hydrophobia also happen. Hydrophobia means they could not able to swallow the water. Why? That striated muscles is going to fully blocked because this striated is a contracted, not release that. So finally, that person going for spasm, he could not able to walk in the nature. So it being like 4 legs is going to moving that. So he could not able to eat a food. So finally, it leads to the mental tension. It leads to the mental tension. Finally, is person moving to the coma stage. It's going to death. Because the entire brain was malfunctioning. The entire brain signal, it is stopped because of entire brain cells are fully damaged due to the replication of the rabies viruses. And also, how it was classified that once the person having the virus, if the person having the rabies virus below the knee, it can able to charge. It can able to travel to the distance by the it will take for example 1 millimeter per day or 2 millimeter per day, 2 millimeter per day. But it can be if it is in above the knee and below the neck, it can be able to travel. For below the knee, it will take 6 months in process. 6 month process. Above the knee and below the neck, it's going further. Below the neck, it's going for 3 months in process. And also near the face, if bite was happened near the face, it will take within a day of 7. That's based on type 1, type 2, type 3. If a person having bite below the knee is a type 1, above the knee and below the neck is a type 2, or near the face is a type 3. How it was classified depends upon the distance towards the brain. And also why the distance towards the brain and the transfer of brain. How long it will take within time of it. So based on that, this is going to happen. How we can control that? We can control by this hydrophobia, photophobia, aerophobia, and finally reach the spasm, reach to death. These are the rabies in human. How can we control that? We can going for the first aid with water. So while washing with water, those viruses, bounded virus in the spot going to scratches, going to wash out. Because once virus was entered to our system by the biting, so immediately that virus, it will be in not a higher bond, it's only in electrostatic interaction. And applying with the high potable water, it leads to the removal of the virus particle. Next, you are going to washing with that soap or solutions. The soap or detergent containing sodium hydroxide, potassium hydroxide as a chemical compound. So that thing, it will be interact with that lipid, what covered with rabies viruses, and the lipid, that entire lipid layer is going for denatured because of the sodium hydroxide. Next, we are applying the Dettol or antiseptic cream. If there are any trace, going for a washout. And finally, going for any other spray or anything, going to everything come out. Even though we may going for the special injections. If a person going for near the bite was happened near the face, if there is no possible to the vaccination, because once the person going for vaccination, it will take minimum 7 days towards the development of antibody, further 7 days for the maturation of the antibody. Totally, it need 14 days. But within a 14 days, that virus will reach to the brain. So that kind of type 3 patient, immediately the person going for supposed to be in what do you call it? Is in a convalescent plasma therapy. What called this is a passive immunization. So the passive immunization, which can immediately relieve that. So then moving for the what do you call? So moving for the active immunization towards the synthesis of that thing. So these are the things available. So how the vaccine was produced here? And as persisted 1907 until December 2004, we are in the production of vaccine that is called a neural tissue culture vaccine. That's a neuron-based vaccines. That vaccine we are supplied until December 2004. As per the guidelines from World Health Organization, that vaccine was stopped. Why we are stopped? Because that vaccine is a neuron-based vaccine, and also for the production of 1 liter of the vaccine, we are going to killing of 40 healthy sheep life. 40 healthy sheep. So that means for the treatment of single person, we need 120 ml. And also it's an unpurified vaccine, and also it will causing 1 in 10,000 to the neuroparalytic diseases. To overcome that, we, the group of scientists, we are developed the vaccine against the rabies called as a cell culture derived vaccine. There's a whole viral vaccines. For any kind of viral cultivation, viral cells, we need a media. So here you can see that pink color is a media. So media is a two type. One is a growth media, another is a maintenance media. Why we need media? What are the constituent in the media? So media containing rich amount of media means it containing essential and non-essential amino acids, and also other factors that the entire thing present that media. And growth media means it contains rich amount of growth promoting factors like phytivin or G protein, entire thing that was supplemented by the serum, like a fetal cough serum or fetal bovine serum, or it is a newborn cough serum. That pH is less than pH is 6.9 to 7.2, and also it's having the less amount of the bicarbonate conditions. And that media utilized for the cells. In our Pasteur Institute, we are using one cell line. There is a VERO, VERO. That VERO cell line. It is derived from African green monkey kidney epithelial cell line. That cell line, it can be easily adaptable, and also it can grow in any type of condition. So we are going to grow in some conditions, like it is a glass bottle, or it is a plastic bottle, or it is a microcarrier bead. So once the formation of monolayer, you can see the monolayer. You can see the monolayer. Here is a spindle-shaped cell line. So once the formation of monolayer, the reagents, that also cell culture, PBS and other things, we have the things. Once the monolayer formation, then that external media, so confluent was reached there, so immediately remove the unutilized media, then we are going to add it with a maintenance media. That maintenance media role is it can maintain the cell line in as such condition without any induction of mitotic cell division, what was done by the growth medium. So monolayer formation. Then we are moving to the next is called as a viruses. These are viruses. And also we have the viruses. That is originally developed by and isolated by the Louis Pasteur. That virus name is PV11, Pasteur virus 11 Pasteur. So we obtained that virus cell line from the Institute of Paris, Pasteur Institute. From that viruses, that is we keep as a mother seed. So that mother seed, we kept in a minus 197 degree temperature using with the liquid nitrogen vapor phase. From that mother seed, we received 2 vials. From that mother seed, 1 vial, we are going to propagate it to the secondary mother seed. From the secondary mother seed, we are going to prepare a primary working seed and secondary working seed. So the secondary working seed, everything we have stored in a minus 197 degree temperature, and the secondary working seed is stored in a minus 80 degree temperature. So the secondary working seed, we are going to the infection. That is called as a virus propagation. So that viruses, we are going to the infection to the monolayer form of virocell line. So once that is a fixed virus type. Okay, another I'll tell that. So virus in two types. One is street viruses, another is fixed viruses. Street viruses mean it is isolated from the animals. We could not able to know their specific function of that viruses, and also what is their incubation period, and what is their infective titer. Everything we does not know in street viruses. But the fixed virus type. Fixed virus, it is isolated. It is carry forward from the street viruses. That is going for the adaptation. That adaptation using with some kind of virus. After the represent the PV11 rabies virus strain, so going for infection. So after the infection, it is a roller bottle culture, it's a microcare culture. So it's going for after the infection. After the infection, we release a harvest. Every 72 hours, that virus, okay, for example, it's a virus, that means virocell line, it's round shaped. So going for that virus going to attach on the surfaces. After the attachment, it leaves their RNA. That RNA entered the cytoplasm. So it's going for transcription translation process. See, inside the cytoplasm, it's going to it's secreted the H protein, M protein, L protein, entire protein secreted inside that. Then inside it's going for formation newly synthesized rabies virus particle. That rabies viruses come out by the budding process. Until if it's a coming on process, that inside that assets virocell line, it is going for intact on the surfaces, attached on the surfaces. There is no damage of the cell line. So only the virus particle is going for release to the cell junction. So the virus, newly synthesized virus particle come out. The come out virus particle and the newly synthesized viruses, it will be in present in a maintenance media. What is floated there? So that media is called as a viral harvest. That viral harvest, what we are moving to do, that viral harvest, we are going to do viral collection by the various period. Every 72 hours, that is a viral harvest 1, VH1, VH2, VH3, and VH4. So 4 viral harvest that is pooled, that is called as a single viral harvest. Then that single viral harvest moving for the concentration, that is called as a clarification. So clarification means, so we want to remove the unwanted material. What is unwanted material? The cell metabolites and unutilized media, and also other traceability. So everything going for removed by the concentration based on the molecular weight. So generally, the rabies virus protein is higher than above molecular weight. So we are the principle called as cross-flow filtration mechanism. You can see the cross-flow filtration is a filtration mechanism. This cross-flow filtration, once passed through by tangential flow mechanism, so the unwanted material going for the filter is called as a filtrate, and the needed material is called as a retentate. That is going for concentration 200 liter of viral harvest. Once we are going for started, finally we'll get 5 liter of the concentrated material of the HVA, that is also corn. So that material underwent for the IPQC testing. IPQC testing means it's a in-process quality control testing. Once that was passed, then immediately going for the inactivation. Generally, we are using with a beta-prepilactone. It's an inactivating agent that was be ensured by the evidence, ensured is using with the animal inoculations. Once the animal inoculation, animal will be in surveil, immediately going for the purification. That purification using with the zonal, previously we are in zonal centrifuge, then now we have the chromatography system. Affinity chromatography mechanism is an affinity chromatography mechanism to give a suitable purification and selection of the fractions based on that antigen content, based on lesser amount of cellular protein, less than the cellular DNA. So after the purification, then going for a formulation to meet. So purification and that entire thing, the quality control tests like safety, efficacy, potency, immunogenicity, that's a quality attribute of the vaccine, then moving to the formulation. So formulation of suitable stabilizer additives, they're going for filling in a filling vials. So if the filling vials coming out as a 2 ml vial, we are going to the filled in a 1 ml of material. Under vacuum condition, it's going for the freeze-dried condition. Freeze-dried. So freeze-drying mechanism, it's a two different cycle. One is a freezing, happening with the minus 40 degree temperature. The liquid material converted to a ice formation. Then we applying with the driving force as a vacuum. Vacuum. What is going to the vacuum? That vacuum to the entire sucked out the residual moisture. So once the entire moistures come out, that only that water all the water molecule come out by the suction of this thing. So only that while protein and the stabilizer only be in form as a button stable form. So as such, vacuum condition is going for sealed and then it's called as a vaccine. So once the production was completed in a production process, we immediately inform to the quality control department. So quality control department, it is in a various type. One is in vitro departments and in vivo department and raw material testing department and microbiology department. So they also taken our vaccine. It's a random sampling through the guidance of QA, quality assurance. So the random sample they are collected, they are going for testing for the vaccine safety, efficacy, potency, and entire things. What we got the results from that, our production, that is IPQC results. If that QC people also will get the same kind of results, immediately that vaccine sample, new vaccine samples are sent to the regulatory concerns. That is the QA sending the National Regulatory Authority. So they are also reassess our vaccine safety, efficacy, potencies by the as per the mandatory requirement of Indian Pharmacopoeia. What we are following that. Once we receive the certificate after passing the test, we are supplying the vaccine through the logistic vehicle. So these are the whole viral rabies vaccine production of vaccine. For the production of one batch, it will take minimum of 8 months along with the minimum of one batch along with the entire test. Okay. So here, some of the flow diagram. So it is a continuation of flow diagram. This is some simplified method. So Vero cell we obtained from the American Deaf Culture Collection. That's also master seed and working seed. PV11, earlier it was the PV11 rabbits virus master seed, originally developed by the Louis Pasteur. That master seed, hot water. Then going for the working seed, then once reach the confluent monolayer, that monolayer going for infection, reach then every third day, every 72 hours, we are collecting the viral harvest, VH1, 2, 3, 4. That is the single viral harvest, then going for concentration beta, CFO or TFO mechanism. Then 200 liter concentrate to a 5 liter, that 5 liter material based on the viral titer. Then moving to the inactivation mechanism. After the inactivation mechanism, a principle, then going for purification in affinity purification. During the affinity purification, we release that. What we are releasing? We are selection of the fraction based on the protein content. Based on the protein content as well as the based on the antigenic content, based on the lesser amount of host cellular protein and host cellular DNA. What is that host cellular protein? What's host cellular DNA? We are propagating the viruses through the Vero cellular DNA. But Vero cell, there's a host cell. That host cell protein as well DNA should not present in our this fraction collection, because if it is there, it leads to the cancer formation. So that's why we are moving in affinity purification. So once it will be in free from the entire thing, then we're pooling and the diafiltration of the entire thing. So once pooling diafiltration is completed, then we for clarification through 0.22 micrometer filtration. Finally, it is called as purified form of rabies protein and ready to formulation. Either it is a freeze-dried or is a liquid-based material. So these are the production process of whole bacterial vaccines. I am going to conclude now. And also there are no such things as a applied sciences. Only applications of sciences by the Louis Pasteur. Thank you. Now it's a time to question hour. I have seen some of the questions in the QA chat box. Okay. I first start with, sir, corona vaccine belongs to which category, which technique? Corona vaccines. Answer live. Okay. Uma Parvati. Okay. So Uma Parvati, You asked about sir, corona vaccine belongs to which category technique? So corona vaccines have different kind of technologies. Whole virus inactivator, what it is called as a Covaxin, and also adenovirus-based expression is a Covishield, and mRNA-based vaccine by the Sputnik, and DNA-based vaccine by the Zydus Cadila, and the VLP-based vaccine also there, and Subunit vaccine also there. So different kind of vaccines available. This thing. So, but India we have Covaxin is a whole virus inactivator, the technique, and what do you call it is in Covishield. It is a RNA-based vaccine and expressed in adenoviruses. And the next is, I thought they got an answer. Okay. And Dr. R. Aluguraja, is there any vaccine for cancer? So which cancer you specify that? Okay. And cancer also we can use with that, what do you call, HPV, human papilloma virus cancer also, cervical cancer also we have. And different kind of cancer, based on the cancer, a lot of vaccines research is going on there. We're not clarifying that which cancers are. Okay. And next is Simi Cyrek. He asked about if a vaccine is not approved by WHO, then what can be said about the efficacy? That means that vaccine should not come to the market, shouldn't come to the public. Okay, if a vaccine not approved by the WHO, then what can be said about its efficacy? So WHO, it is not a direct role. That WHO, it will be an full authority based on the particular national regulatory results and other things. Okay, and another question, Dr. R. Abbas. Today's lecture is related to recent trends pandemic period. As a researcher, which vaccine is you recommend for the COVID? As per my concern, is a whole virus inactivator. Why I say a whole virus inactivator means, so here that entire virus particle is an entire virus was fully propagated. So after fully propagated only with a concentrator and an inactivator, that means contain 100% of the viral particle things. That's why my suggestion, as per my as a vaccinologist, it may going for the whole virus inactivated things. If going for mRNA, mRNA is related to some what is spike protein and other kind of things. It is focus on particular thing only that. Okay, and Parani Selvam, whether all vaccines are tested in human body before injected, especially yes. Then only it can be coming to the already clear that I thought at that time 7-8 that time you were not clear about my slides of vaccine development page one. That phase one is a trial. Phase one, during the phase one trial, that vaccines are instructed in human bodies. Yes. And another one, there is no name is there. He mentioned that what is that? Galaxy J7 Next. Sir, for COVID vaccination, same dosage is given in respective ages. What is the reason that? Yes, that is on a phase one trial, escalating of the doses. What are the results obtained? Whether it's a 0.5 ml or 1 ml is it can be that time is decided in a phase one level. It is not. There is a depends upon the phase one results. So that way that was given that. And again, next, Pioneer Selvam. What is the position of our country in vaccine development? So, okay, vaccine development. What is the position of our country in vaccine development? So, sir, okay, just you go through the Ministry of Health and Family Welfare. So they also detail about only India we have the 2 vaccines are released now. One is Covaxin, another Covishield, another one is Zydus Cadila, another one is Indian immunological vaccine also in trial, and Arabian form vaccine also in trial, and Cadis another vaccine also trial, and biological E the vaccine also trial. You can get much more information from that. What do you call in the our ministry website? Okay. And the next is Galaxy Days. Why is at all of us don't have the same symptoms after vaccination? That means symptoms is what I can't able to that questions. Why as it all of us don't have the same symptoms? It is not a symptoms. Okay, symptom it is an what I call it is an after vaccination. That's why there is a depends upon the person. So that depends upon the immunogenicity and it depends upon the what I call depends upon whether they're having the comorbidity. So comorbidity means whether they're having any kind of problem like diabetics or pressure or kidney problem, that kind of things there. Okay. And again, sorry, asking my question. Why some vaccines given in the form of drops? Yes, that is called as a different kind of route of injection. So first, my slide is a first title is a vaccine, either it's intramuscular or intraperitoneal, or it is in this thing, root or nasal, or going for pulse polio vaccine. Because that is a good route of injection. Even newborn babies, up to the child, we cannot able to going for the intramuscular vaccines. The polio drops, it can be easily, they also, the injectable, that's going for easily to that digestion tract. It is not lose a specific activity. That is a newer, and in future also we'll get a lot of vaccine in here and drop formation. There is a oral based vaccination. Only two vaccines in the world. One is a polio and another one is rota. It is in what do you call it? It is in oral based form. And another today also some introduction. There's a nasal based vaccination also there. It's a newer development. Okay, and the next is Dr. P. Veerasamy, respected drug allergy person take COVID vaccine. If yes, what type of hypersensitivity reaction occur? Sir, your question not clear to me. Drug allergy person, which kind of drug? Persons take COVID vaccine. If yes, what type of hypersensitivity reaction going to occur? A drug allergy means whether it's based on antibiotic drug allergy or it is in steroid-based allergy or different some allergy persons. So what type of hypersensitivity reactions occur means I'm not sure. Sorry, sir. This one. Okay. Thank you. And another is biology T. Which type of vaccine burst for COVID is Covaxin, Covishield? So already told that means any kind of vaccine, it should be in the whole virus inactivator. Because that kind of vaccine only out of 27 vaccine, whole virus inactivated type of vaccine, that points only in major amount. Because that contain enter whole viruses are activated. Viruses. So that leads to the good. And again is, sir, do the cervical vaccine is safe? What do you call Dr. M. Bachira John. Sir, do the cervical vaccine is safe? In what age should be administered and how many doses? In US, those girl child after the age of 12, they're going for vaccinated with that way. They're mandatory. But India, still it is not mandatory. So those after attend the puberty, that is advisable that way. Okay, Professor G. Haribhaskar, sir, what are all the precautionary methods before taking vaccine? You should not have cold or fever or any kind of comorbid things, and also any kind of allergy. So these are the things you can follow that. And also, once going for vaccinated in that area, it should be clear and it is not affected with or any wound or other things. The other things, okay. Where we and Paniculum, where we applied gene-based vaccine and where we applied virus-based vaccine. And depends upon the virus, depends upon the, it can be clear of efficacy, safety, potency, and immunogenicity. So these are the things will be in that factor to decide the gene-based vaccines or need or virus-based vaccines or need. So gene-based vaccine, virus-based. Okay, that means it will be a virus inactivity-based vaccine. Yes, okay. Whole organism-based. And the next, Josephine Rebecca. Good evening, sir. Which is more effective DNA vaccine or normal vaccine? Still date, only one vaccines are it is in DNA based things, but none of the other vaccine it has got a very good efficacy. And as per my knowledge, but other vaccine is effective out of 27 vaccines here. And Sylvia Ramasamy, she written, what is that? Recombinant. Oh, some typography error. Recombinant vaccines are produced against COVID-19. Yes. If you want much more detail, just go to that WHO World Health Organization, WHO site. There's a landscape, LAND, landscape of COVID-19 vaccine. In that also you'll get which country and which develop the vaccine based on the platform, either it's mRNA or DNA or VLP based or recombinant based. All the things is available there. Still now there is no recombinant based vaccines or come to the field now. Okay, that may be clear. And next is Mariam Janbahar MS. Since pneumonia and coronavirus have more or less symptoms, can the pneumococcal vaccine give some percentage of protection from coronaviruses? Okay. Since pneumonia. And pneumonia also caused by either bacterial pneumonia is there and also viral pneumonia also there. But symptoms mean less same symptoms, like it being both are affected the upper respiratory tract infection. But pneumococcal vaccine, pneumococcal pneumonia vaccine, it can be in what do you call it? It can be protect against a particular virus thing. And also pneumococcal is a conjugate based vaccine, and also it is a conjugate based vaccine. Once you're going for that coronavirus, it cannot be converted in the immune system. Okay. First, a clear manner. My second slide, I told that. What is that? If a vaccine, what vaccine we are vaccinated, that the antibodies are delivered against a specific, that antigen is going for delivery of a specific antibody. That antibody, it can be interact with that specific and antibody only. But here, pneumococcal vaccine is going to cause a pneumococcal antibody. That antibody, it can be interact only pneumococcal virus. How can it be possible that? Okay, I thought maybe clear now. Do the D-dimer test and antibody test taken before the corona vaccination protect us from vaccine failure? I'm not sure, sir. Listen, I don't know this thing. Sorry. Okay, why different countries are using different vaccine for coronavirus, and any research has been conducted so far the country vaccine giving higher percentage protection from the coronavirus than the other? Why different countries are using different vaccine for coronavirus? Any research has been conducted so that the country vaccine giving high percentage? Yes, sir. If you need much more information, already told WHO World Health Organization, just go to the WHO website and also the landscape. In the landscape and which country having the which kind of vaccines now, and also which stage, the page 1, page 2, page 3, and also on that page 1 or page 2 or page 4 condition, what results they are obtained, and that results, the entire results they are in published in a leading magazine like Lancet and JAMA and different magazines. You will get much more clear that's answer. Thank you. And next is anonymous attendee. What are the mild to moderate side effects of the COVID-19? Mild to moderate side effects. COVID-19 or COVID-19 vaccine? So answer is not clear. Again, the same attendee to answer is not clear. Okay, sorry, question is not clear. What are the mild to moderate side effects COVID-19? Is this vaccine? So it depends upon the person. Okay, and so how long will take build the immunity after getting COVID vaccines? As I already told, if a person, if it's a general thing, if a person going for vaccinator, 7 days needed to the development of rabies vaccine. Development of antibodies. Further 7 days needed to the maturation of the antibodies. Totally, it has a 14 days to the development of antibodies. Okay, any kind of vaccine. And the next is for dog bite. Is it correct to wash with salt water or water crystallized salt? But as a dog, as a rabies vaccinologist, any kind of normal water, just don't go for that salt-based water. So why we are been washing with water means, so once the bite was happened to the person, and that bite area, that rabies virus is going to bind to that particular area, into the muscle cells, through the electrostatic interaction, only the weak one. That weak one, we are going to the releasing, we are going to the remove the weak one by the applying the water wash. Okay, if you are moving for salt water, what is the use of that thing? Okay, we just on the water and going for the what I call it, you can wash with soap solution. Soap it containing sodium hydroxide as well as potassium hydroxide. It can be digested to the lipid membrane. Okay, good. Next is Dr. D. Brighty. How can we distinguish between active and passive immunity? Active immunity means once you are vaccinated, you will take development of antibodies. So passive immunity means if for passive immunity means that person, for example, if you're type 3 patient of rabies, if you're going to give the vaccine, it will take minimum 14 days. If it has happened to the near the face, so there is no possible for the development of vaccine within a 1 day. So that's why the passive immunity means already secreted antibodies, and also purified form is that I'm going for immediate trigger immunity, neutralize that antigen what you are encoded through by that. So we can use the passive immunity. That is a passive immunity, already secreted antibody. And also in COVID, we are using it convalescent CP therapy in a first way, convalescent plasma therapy there. Okay, and another anonymous attendee. Bureau takes 8 months to 1 year to. So bureau-based vaccine, any kind of vaccine, it will take minimum. It will take minimum to the 8 months for the entire certification. That is 8 months to 1 year. Anyways, then how about COVID vaccine? But COVID vaccine, for COVID vaccines, for the whole virus inactive, it will take minimum of 8 months based on the certificate by the approval authority. Which vaccine would you suggest for COVID-19? My best one is any vaccine should be in whole virus inactivator. Why efficacy is different from coronavirus vaccine differ? Why efficacy of different coronavirus vaccine differ? See, Anbaragan, what do you call it? Your question is not clear. Can you type later? Okay. Why efficacy of different coronavirus vaccine differ? Okay. And also next to that, okay, just Rebecca. Okay, the next is Jayanti Jayaraman. Okay, excellent presentation, sir. Why vaccine being success in research but reacting in people different? How could we assess? Vaccine being success in research but reacting in people different. But depends upon the personal. Okay, depends upon their personal immune response. How they are triggered that. Okay. And Tilakam Gopalakrishnan, why do kids need vaccine for a disease that's been eliminated? Okay, that's been not eliminated. If that disease going for fully eradicated only, we need that. Sometimes the vaccine reverted based disease also may happen. So that time it been a higher amount of higher amount of pathogens will be there. So that's why if a vaccine eliminated means we can continue. That shot will be reduced. Previously, once vaccine was there, we're going for in a 3 shots, 4 shots. After the elimination, we'll get only in 1 shot or 1 shot, and also reduce the time period. And the next question is Taju Syed Ghani. In India, having 2 vaccines against coronavirus. My question: which vaccine more effective? Already told, whole virus inactivator. And Dr. Hirilmudi Sophia. Why there is high expectation for Sputnik based on that media? Okay. Good evening, sir. Kadiresan, may I know why RNA vaccine is highly unstable? Why RNA vaccine is highly unstable? In your question, you have the answer. That is the RNA vaccine. And D. Purnima, in CPR test necessary for corona vaccine. So, who has to take the test? I am not sure. I don't know the answer. Sorry. Okay. And can we, Dr. A. Kanagavalli Pun Vetrivel, can we prevent the death rate after vaccination for cancer, especially blood cancer? Okay. Can we prevent the death rate after vaccination for cancer, blood cancer? But still, as per my knowledge, there is no proper vaccine against the blood cancer. Okay. One of the Nobel laureates suggests to take vaccine. If so, what is your opinion? You may ask Nobel laureate, man. Okay. I thought I not know that answer. Sorry. And also Dr. N. Vijay Samundiswari, sir, booster vaccine won't bring any impact of our transformation. But the question is, a booster vaccine won't bring of any impacts on our transformation. No, answer is no, ma'am. And also, Arokya data. Why WHO has not recommend Covaxin? But recommend Covaxin, our national regulatory authority. So based on that emergency aspects, it will release that. And also they got, okay, still there is no vaccine-based death was not happen. So that's why they released that. If you need much more clear from that, you can go with our Minister of Health and Family Welfare. And they also having why the vaccine was released. You will get much more detail. And the next is Galaxy A31. Good evening, sir. Really great explanation. The question related to presence in a COVID-19 vaccine will make any side effects. Depends upon the person. Okay, it depends upon the person, and it depends upon the time how he is going for vaccinator. And as a Karikol Raj, can it possible to produce COVID vaccine at your institution? Yes. That we need. We are specifically for the DPT group of vaccine. Previously for the rabies vaccine. Now we are confident that things. That also we are approaching the ministry. Other things there is going on. Everything depends upon the ministry decision and then government decision. We are moving to do that. Okay. And another certainly how microbiome will associate with the coronaviruses. Mycobacterium tuberculosis and coronavirus both are common infected areas called as upper respiratory tract infection. And the next is Dr. C. Malathi. Out of 2 vaccine, okay, already told that one. Any vaccine for leukemia, you can, I don't know, I'm sorry. Dr. C. and Anodre, which one is best vaccine against what, sir? And Parimala, many vaccine came for COVID-19. Did all the vaccines generate same immune response? All the vaccines are regulate the immune response depends upon the personal things. Okay, person, how they have their immune response. That things. But that means vaccine constitutes what we are vaccinated. It can elicit to the antibody against the antigen. Okay. So either it's a RNA based means it against that RNA. DNA based against the DNA. If protein means that. If whole virus means that one. Okay. Ms. Parimala. And Rajeshwari Bhavana. Sir, how many gap can be there? Covishield vaccine, first and second. It's not a gap. It is called as independent period. The things, but still, any kind of vaccination, the booster dose, any kind of vaccination booster dose will take minimum of after 12 means 1.5 month. After 1.5 month. And also, Dr. T. Subha. I want to know whether the child will born. The doctors put rotaviruses vaccine. Like Covishield, other vaccine will use it for enough. I want to know whether the child will born. The doctors put rotavirus vaccine. Like Covishield. Yes, it will come in the near future. Rajendra and Raman. Coronavirus affected human beings only. Okay, that's a good question, Rajendra and Raman, sir. Coronavirus affected not only as a human being, it was it was originated from that animal. Okay, corona vaccines are veterinary also available, sir. Now only it's affected to the human being, now it's come out to the doctor. Okay, and Kanagasudha Sujata, sir, why Covaxin is not accepted by WHO? You will get information from the WHO. This is on the right platform to here. Within Oscar Navas, if 2 doses of vaccinated human, again how corona reported, whole virus modified vaccine now available. If 2 doses vaccinated in human, again how corona reported, whole virus modified now available. 2 doses vaccinated, okay, what kind of vaccines, how corona reported? That is a vaccine reverted diseases for your answer. And another question you asked that whole virus modified, it's not modified, it's an inactive vaccine now available. Yes. Fatima Safir Farooq, 8A. After taking vaccine, why some people dying from COVID-19? Which vaccine? Whether maybe they're having the comorbidity, like and also their lung functioning, and also their receptivity, and also other kind of factors or things. And Dr. Jimsi Asha, do you advise any kind of checkup before COVID vaccination? Yes, always. That should be disease-free status, and also should not having any cold and common thing, and also should not take any kind of unavoidable foods like அவங்களுக்கு ஒத்துக்காத food. What are things there? No, we could not able to avoid that things. Why means any kind of vaccination, our body should be in kept in 37-degree temperature as well as to our immune system that boost up the cells. That's why it is that. And Uma Parvati again. Uma Parvati. Okay. Why RPCR distinguishes normal cold for corona? I'm not clear with the question, Uma Parvati. Okay, next. G. Ratika. Infected person advised to take vaccine only after 3 months of recovery? Yes. Because the infected person on the 0th day, they have that same kind of rabies, same kind of coronavirus present there. So once that trace always will be in up to 70 to 80 months there, and also it be in a bigger amount, and also to once going for vaccinated, it can be what do you call it, diminishing the immunogenicity effect. That's why they advise 3 months that one. Once the recovered corona patients going for the vaccinated, that antibody it being sent up to the lifelong. A paper was published in Lancet. Okay. Logeshwaran J. Hello, Mr. Logeshwaran. Can you provide any significant vaccine for black fungal attack? Okay. Still, there is no vaccine against any kind of black fungal. So here, there is an opportunistic fungi. What you are asked to know, opportunistic fungi. Why it was coming out? So during the person having that COVID infection, so automatically that coronavirus is going to bind in the ACE2 receptor. So immediately, how our immune system is going for detect that, it may be happen for that something is coming out. So there's going for rendered towards the cytokine secretion. So immediately the cytokines are much more secretions. So that what we are using, we are going to elevate the cytokine secretion. They're going for to in fact to close that one, to diminishing that one. They're using with some steroid. So the steroid is not only for cytokine forming blocking, the entire immune system going for block. So that also one reason going for development of opportunities fungi. So opportunities wherever it come, where it go. Nothing to worry about black fungi. And also, you can also see the black fungi. One in your home, you kept any bread and bread material. It is not in use for sometimes. After 7 days, what kind of things you observe that? Just small mold extract black. That also black fungal. Okay. So and black fungal means is a opportunities. Wherever there is grow, it can go out. Okay. Still, there is no vaccine against any fungal. Okay. As per my knowledge. Okay. Anonymous attendee. Is the vaccine safe for epileptic patients who is taking medicine? You can consider with the doctor because any kind of medicines, it also diminishing. That means diminishing it reduce the vaccine efficacy. So that's why some of the drug. For example, in some of the outpatients, they are going to blood thinner based drugs. They are going to take. If they're going to the vaccination, it leads to the somewhat of a blood bleeding. I can consult with the doctor. Thank you, anonymous attendee. And Bhavani Karthikeyan, which one is a beat? A beat or best? Okay. Okay. Best one is Covishield. Okay. I don't know. I will not tell the name. One is a whole virus inactivated vaccination. Best. And also another thing, anonymous attendee. Why there is a confusion giving second dose? We studied immunology book after 35 dose. Okay, study very good. Immunology book depends upon the vaccine. Any kind of vaccines going for immunization, it will take after the 3 months is good. 2 doses. Okay, another thing for tetanus aspects, every 5 years the person is going for vaccinated. Why? That also booster dose. That also studied in immunology book. Depends upon the vaccine. Okay, anonymous attendee, you kindly text your name. Dr. S. Sumadi. Yes, sir. Thank you. Okay, answer பண்ணதில்லையே. Dr. Mary Halitaye. Can vaccine cause any allergy reaction in human? It may be in some of the vaccine. So that kind of things you can address during the what do you call it? It is in phase 2 aspects. So if any allergic was happen, that type of vaccine is not come to the market. Okay. And also next is Tilakam Gopala question. Can the ingredients of vaccine cause allergy reaction? Yes, some of the ingredients like preservative like thiomycin, it causing autism and other thing. So that kind of thing should be in a lesser amount, depends upon the person, and also that approved that is a 0.001% only. Something it maybe happen to that 1 in 1 crore people. And Govindan Veera, why are some people affected coronavirus after vaccinated? There is a vaccine reverted coronavirus infection. Okay, Mr. Govindan Veera. Again, Venkata Subramanian. After taking vaccination, there was no fever or body pain for me. It means whether the vaccine is working or not for me. Yes, vaccine always working. No fever means that means your body is out to the vaccine. So good. And the next is Dr. C. N. Anadure, former Chief Minister. Which one is the best vaccine? Whole virus inactivated. Thank you, Mr. Dr. C. N. Anadure. And also another question, anonymous attendee. An Italian scientist tells we are taking vaccine our body as a virus. Itself, then there is a chances for we have transformed viruses. I am not able to answer your anonymous attendee. Kindly text your name. Dr. Shanmugam Yan. Sir, could you able to produce vaccine at your pace? Yes, depends upon our ministry decision. Feedback link, sir. You can also your organizer. Okay, thank you. And Prasanna Kumar, sir, I don't know what is a whole virus inactivated. Can you tell? Just you can get the information from the vaccine. And okay. And also Corona, Dr. Al Suji. Coronavirus affected several ways. Why? Is the virus created laboratory or not created? I don't know, Dr. Suji. Several waves means okay, it's not newer, Dr. Suji. Previously, we faced that means we are not based on the history, based on the pandemic. We already faced the plague is a plague disease also there, and also what you call influenza also there, and also recently. So different. It's a wave means it can be spread through this. So once it's a pandemic, it will come. An epidemic like that, no, it will come not only for corona. Not a several waves there. Just to go through our Google also. What are the previous waves against diseases? Okay. And next, Dr. K tells how to test immunity level against what vaccine. If going for rabies means you can get your serum. That's him going for test in at antibody level with repeat testing. That things. That's not clear in the question, Dr. Kalaichili ma'am. And the anonymous attendee. Even after vaccination, people get corona. Then what purpose of vaccination? That means vaccine reverted that one. Next is anonymous attendee. Oh, okay. Next is it's safe COVID vaccine after total hip replacement surgery. Consult with your doctor. Okay. Rajeshwari Bhavana. What is the best vaccine for Delta virus? Delta means those from India. Best vaccine means any kind of alpha, beta, gamma, delta. We may go for that whole virus inactivator. And the next question from the Dr. A. Kanagavalli Pun Vitrivel. A person with high parathyroid hormone secretion can take any one COVID vaccine. But for the parathyroid hormone, you are taking some of the medication. That medication, it can be what do you call it? It can be diminishing. That means diminishing your antibody level. So that's why you can consult the doctor. And also next is Arogya Anbaragi J. Excellent session, sir. Nandrigal Mikke. My question is, I have 2 kids. My elder son, the vaccination card which was given to the follow as less number of vaccine, but my daughter was given to the more. Does the number make any difference both their growth or immunity? I have 2 kids. For my elder son, the vaccination card which was given to follow has less number of vaccine, but my daughter was given with more. Does the number makes any difference in both growth and immunity? But that means depends upon the year. Previously it was in vaccines. There is no more number of combination vaccines. Only diphtheria, tetanus, pertussis. Nowadays is a 5 shots or 3 shots, 4 shots, different vaccines are available. So that is going for single shots contain 5 different shots. So that way reduce the number. Okay, Arugya, ma'am. And Surya Kala. Is there any death reported due to the general vaccination? Vaccination against which disease? Can you text that? Dr. Vijaya EMGYWC, sir. Now, COVID has spread to animals. So, whether it spread from animals to human will cause risky. COVID vaccine first started with the SARS and MERS. MERS, okay. It's starting from, it's a zoonotic. It's from the zoonotic, sir. So, G. Aribhaskar, sir, it's a mandatory for CRP test. Sir, I don't know the CRP test, Aribhaskar, sir. And what are the side effects of this vaccine? Can you share? Can you give that what type of vaccine? And also again, Josephine Rebecca, okay, Italian scientist. You just I don't know the answer, man. And the AVP Karthikeyan. Which vaccine did put against COVID? Already told the answer. GNS Kannan, CH Chennai. Is it good to have side effects of COVID vaccine? Okay. Is it good to have side effects of COVID vaccine? Is it good to have side effects of COVID vaccine? Depends upon the person, sir. Okay, Karikul Raj. Why vaccine producer company charging different prices, private and government? Okay, there's a production cost and also their investment. That is a company policy. Thanks. Okay. This is not related to our presentation. Okay. And Dr. P. Rathinam, GHCB, sir, how did SARS virus dissipate before finding vaccine? So not only SARS, a lot of diseases came out. COVID disappeared, and SARS. That means SARS means is there? Is a mortality? Is a morbidity rate is lower when compared with the corona, and also mortality rate is lower. So that's why there is a disappear, sir. And also it is not declared as a pandemic, because it is not affected the entire whole. It will be in some of the few area only in affected, Dr. Rathinam, sir. And next is Pioneer Selvam. Is there any relationship between vaccine and the climatic condition of the country? Is there any relation between vaccine and the climatic condition of the country? Not clear your question, Mr. or Dr. Pioneer Selvam. Any relation between vaccine and climatic condition of the vaccine? But any kind of vaccine, any climate condition, if you are going for vaccinated, our body is maintained the temperature 37 degree. As a 37 degree, it's going to activate that. If you are in Iceland, if you are in Africa, anywhere, or if you are in any desert, okay. For that, your questions, I'm not. This is my answer, sir. And next is Dr. Maria. Does the saliva of rabbit dog bitten person contain rabies virus and could be able to cause rabies another man? Yes, possibly. If that person, that man having the bite, that having the small wound, if the small wound was, if the saliva is going to contact the small wound, it leads to that transfer of rabies viruses. Okay, good. Dr. Indra PM, is the vaccinated body part became thicker after vaccination? Yes, that is okay. That is called as because the vaccine was stored in plus 40 temperature and going for vaccinated in that particular area. It's going for that. It will be in what do you call it? It will be in something. It's going for sustain release that. So it will like thick, not thick. It will be in it will be in like something. So it is not in on the area only. It will be in what do you call like harder. When going for it will be in sustain release that. After releasing, it leads to that will be normal, actually. It is general for all vaccinations. Veeramani, what are the animals are carrier of rabies viruses? I detailed in that slide. So, dog is a 95%, and 4% the cat and jackal and sheep, or there sometimes the camels and also the raccoon and other things there. Animals are link. What link? I don't have a link. Veeramani and other. I'm going to complete. And also again, Raghuraj Rajaguru Pugalindi, sorry. After vaccinating dog for rabies, will still rabies will be in the dog saliva? If your dog will be vaccinated every year, that those viruses from the dog saliva, it will be in inactive mode. That's why it will be for going for vaccinated dog every year. And Dr. Yelmudi Sophia also another question: why there is high expectation for Sputnik? Because of media. Okay, anonymous attendee. Any articles or intelligent scientists tell coronavirus is from Wuhan, so the clinical made scientist virus? It is not relevant question to here. And the next is Dr. Indra PM. What is the difference between? Okay, that's a whole virus inactivated. It's from adenovirus express RNA. And Dr. M. Shiva Shankari. My husband take dog bite injection till 29 April. When will we put corona vaccines on? Okay, 29th April. Whether it's a first dose or it's a booster dose, but it's not problem. You can consult the family doctor. If already 29th April, no, so no problem. But anyway, you consult with the family doctor if we are not having any kind of comorbidity. Dr. Rajeshwari K. Is there any issue in taking Covaxin during menstrual period? Thank you so much for nice and wonderful presentation. And Dr. K. Rajeshwari. No issue against taking any kind of vaccine during that menstrual period. You can take the vaccine. Okay. And Dr. H. Mohanavalli. Is it effective when we take second NND dose after 3 months, the first dose of COVID vaccine? Yes, any vaccination. So already I told the example about tetanus toxoid. Every 5 years, the kids are going for vaccinated up to that 18 years. So no problem that. And the next is anonymous. Good evening, sir. Like coronavirus prolonged mutation of H1N1 spread through, are it's possible? Yes, there is a strain variant going to happen. And next question is Dr. N. Kavita. Respected sir, if the kids who are vaccinated for all diseases up to 2 years can able to prevent from corona by Dr. N. Kavita. Okay. APA College Culture Prani. Okay. Respected sir, if the kids who are vaccinated for all diseases up to 2 years can able to prevent from corona, yeah, it can be. And next, Dr. S. Chitra, excellent presentation. How long after second vaccine? Second dose vaccine are you immune? How long after second vaccine are you immune? Second dose. After that, any 1.5 month. And Param, can we go additional vaccination of a different brand after administering one brand? That is called the interchangeability vaccine. Still, India, that is not done really the study of that things. But previously, it was this kind of interchangeability vaccine. Param, interchangeability vaccine also happened in the whole wholesale pertussis vaccine along with the live pertussis vaccine also happened, and anthrax also happened. But still in India also, there is no much more data against that interchangeability vaccine that one. And next is Unburden C. Is there any test needed before taking coronaviruses? Sir, I'm not sure that. And another anonymous attendee, is that safe to take COVID vaccine after total hip replacement? Sir, please, you can consider with your doctor, sir. And next, Dr. V. Kritika, DKM College, Vellore. Good. Whether the mRNA vaccine after alter our genome. Okay, that's a vaccine. Okay, that's a vaccine going to elicit immune response. Is there any chances if vaccine that mRNA it won't be converted to a? It is not active mode. It's only mRNA based. What do you call mRNA based antibody only going to produce that. It won't be auto. Okay, Kritika ma'am from DKM College, and also Sen Tamil Prabhu. Is there any vaccine for bird flu? Yes, available. Not in India. Is it advisable for people with allergy to tablet like Droxel to COVID vaccine? Okay, sir, you consider with doctors, please. Dr. S. Bhuvaneshwari, Dr. S. Bhuvaneshwari, GVG Commerce. Okay. How long period vaccine is function in human body with vaccines more? Okay, I'll give a clear that for the newborn baby born, immediately they're going for vaccinated BCG. That's a single shot that that elevated antibodies should be in up to their lifelong. But in case of DTP, DPT group of vaccines, every up to 5 years going for booster doses here, then only it got antibody level. And also for tetanus toxoid, we need every 5 years going for vaccinated. And measles, mumps, rubella, we are going for 2 shots of the vaccination. So it depends upon the vaccine. And also, Mariam Jenner Bauer, MS, can you specify the importance of rubella vaccine for females and can it be vaccinated 15 to 20? Can you specify the importance of rubella vaccine? This is not related to this thing. If you need, just you text me, I will answer that question. And Veeramani, what are the animals are carried of rabies? Well, already I told that 95% is a dog, street dog, sometimes a pet dog. Dr. K. Subramanian. Why doctors are advised to take second dose of the COVID vaccine after 40 days? Better. Is it right? Yes, sir. Fatima sir, thank you very much, sir. Myself, Dr. Nantin Forik. Thank you, 8A. Okay. And also another one, Dr. Jeba Jasmine. D and Jeba, what is the difference between, already told, whole virus inactivated? It is mandatory to take vaccine. Which vaccine? Already told. Is whole virus inactivated? And Dr. Smati is asking about, can we follow second dose for animal also? Yes, absolutely. It is against, I thought it'd be against the rabbits. Every year the dog should be vaccinated properly. That's a boost doses. Can you follow 2 dose procedure animal also? Yes, ma'am. And the Paratini question, sir, after taking 2 dose vaccine, some people affected the COVID-19. So COVID is COVID, sir, not COVID. COVID. Okay. As COVID. That means there's a vaccine. That is a vaccine reverted. It is curable, sir. Nothing. And another is 09C6 is something. Sir, I want to know the timeline to predict we are infected corona, how safe our life. Sir, I want to know timeline to predict we are infected by corona and how to save life. So based on that biochemistry marker, a lot of kits available that. And again, it's Dr. Ailmadi Sophia. Does COVID patients need vaccine after recovery? Yes, after their 90 days. Again, start after taking 2 vaccines, some people affected. Okay, already told, answer that. Dr. Venkatesh Narasimha Pandian. What is the difference between so the things, it is not related to this question, sir. Already told. So whole virus inactivated, sir. For the second questions, I will not answer, sir. And another nice information, sir. If vaccination, sorry, 1 minute. Oh, nice information, sir. If vaccination is secure or not. Yes, vaccination always good. It can elicit immune response. And next is Kalachelvi Selvam. Sir, does vaccination have any effect on people having breathing problem? Vaccination have any effect on it? You consult with your doctor, please. Sir, after taking the vaccine and affected by COVID, second dose is necessary. After taking the vaccine and affected by the second dose, yes, necessary. After recovery from your recovery from that COVID, then after 90 days, you're going for vaccination. Okay, Jayanti. Okay, Jayanti. Kalpana Tawasik. Good evening, sir. Wonderful informative session. Why diabetic patients are affected more by COVID? There is a comorbid infections. So their sugar level, it is an elevator, and also anything, there is a comorbid means our body, it is not 100% functioning of the immunity. So we are taking like diabetic as well as that, what I call it, is in pressure that. So for this thing concern, there is a reason, sir. Dr. C. Ratnam, thank you, sir. Can take, okay, Dr. Devamani, COPD patients can take vaccines or not? So you can consult with doctor. COPD patients is not, we can take a vaccine, but anyway, consult with the doctor. And also, I have taken the first dose of Covishield vaccine. After how many days, second dose of Covishield vaccine should be taken to me? Dr. C. Maladi. I can take after 2 months. J.H.T. Saira, sir, no question. Why? And also Vijay Kumar, Dr. M. Vijay, preventing corona, I already told. And Jayashree Sairam, thank you for your patient answering to all questions. I could observe the amount of anciently among the learned group. How is it difficult for you all bring in awareness among the Indian mass? Hats off to you, sir. Thank you, Jayashree Sairam, ma'am. Anaxity, okay. And Shyamala Gauri, sir, I already affected corona last month. When I take the vaccine, after the 90 days, then only you will get antibody, or that antibody will be in present up to your lifelong, as per the recent paper published in Lancet. Serology test is okay, sir, regarding what, Kalachali ma'am? Can you clear that? Dr. What is the difference between already told? And another, thank you for patiently reading and answering all the queries. And next, AS Ganga. Whether it's allergic person take a vaccine depends upon your allergic, and also consult the doctor. And Indra PM, can we take the vaccine if planning for pregnancy, sir? Can you take the vaccine if planning for pregnancy, sir? Okay, but you should consult with your doctor, because during pregnancy only you are going to take some of the tablet, like other thing. You can consult with your OG, gynecologist doctors. But my answer is nothing to worry about that, because during pregnancy also, the pregnancy mother is going for vaccination for TT on the month of 5 and 7. That thing also there, so you can consider with that. Thank you. And also that, what I call Rajeshwari Bhavana. Sir, I have taken injection on the 13th March. After that, I had some problem, and my doctor advised to take second dose. Can I take second dose? Rajeshwari Bhavana. Sir, I take injection 13th March. After that, I had some problem. Advised to take not to take second dose. Can I take? Whether that entire problem going to cover 13th March mean, but anything. If you're going for vaccine after 3 months, also it's not a problem for second dose. And next, Dr. S. Praveen Bono. It is essential for diabetic to take CRP test. I'm not sure, sir. Please, sorry. Anonymous, sir, what is the difference taking vaccination or 2DG medicine produced by DRD? What is the difference of taking vaccine? Okay, that is a different concept. Of vaccination means it will going for development of antibodies level. And 2-deoxyglucose is a medicine. It's sometimes it's a proxy glucose molecule. That means the coronavirus is going for taking the glucose, taking the much more of glucose. So that's why the entire glucose is going to get down. To avoid that, to mimic a glucose molecule is a 2-DG. That is a different medicines. That is the entire different. That's a drug based. And Jayajit, thank you for your patient answering all questions. I could observe the amount of anxiety among the learned group here, how it is difficult. Thank you, ma'am. Rajeshwar, sir, can you share your mail ID? Sure. And also, hello, sir, 293AE746. Hello, sir, I have a quick question. Hello, if I take a Covishield first dose, in second take, yes, that kind of things. Okay, that is an interchangeability of the vaccine studies. Still, it is not conducted in India. If you're going for whole virus inactivated, then going for booster dose as a mRNA or RNA basis, still not conducted in India. That is not advisable. If you get a good, what do you call it, good type of data only, then we moving for that only. And also test immunity level serology test. It's a mandatory, somewhat this thing, ma'am. Depends upon the person, Dr. Kalachali, ma'am. And also Dr. Jagadish, sir, after taking 2-dose vaccine, it's a wear mask. Yes. That means why we are taking the risk for this? No. But in our India, some of the people not wear the mask here now. We can wear. And also another thing, one statistics I will share you. Previously, a lot of people are going to the hospital regularly for that affecting cold and other things. Okay, once the mask was introduced in the world, that kind of problem was not happened here. Cold and cough, everything, because they cover our nose and other thing also there. So that's a good. In future also, we always wear the mask. Good, Dr. Jagadish. And another is Dr. D. Uma. Those who are taking medicine for TB and chronic disease can be a vaccine for COVID, but TB and other things, you can consult with the doctor, ma'am. That is not advisable. Anything, consult the doctor. That I answer all.
Yes, sir. Thank you, sir.
Thank you, sir. Again, some of the questions, some of the raised their hand, I could not able to see that, sir.
You can post your email ID, sir. They will ask you. They will.
Okay, sir.
The chat session.
Okay, sure, sir.
That's enough, sir.
Okay, I will share. 1 minute, sir. So where are you going to? Post my email ID, sir.
Chat, sir.
Chat. Chat.
Chat box.
One minute, sir. One, sir. Okay. The same that Q&A box I will answer, sir. Okay. Sorry. Okay. The type answer. Somebody asked, no, send. Taking clues something. Okay, I will answer here. I could not able to type here, sir. Our Q&A session. Where shall I type, sir? I could not able to reach that one.
In chat session, sir.
Chat session.
Your mail ID. Just now I saw your.
Yeah, something.
Right.
Okay.
I will type. You tell the mail ID, sir.
SEL.
SEL.
JAG. JAG. 2005.
2005.
Okay. At gmail.com, sir. Sir, okay. Yeah, I got. No.
Finish, sir. Finish, sir.
Okay, so one minute, sir. SEL JAG 2005 at gmail.com. And I also drop my my number also 9486081. Okay, 9486081. Okay, sir, shall I put privately or send all?
Send all, sir.
Okay, sir. Type answer. So just you check with the answer, sir. You can send to all, sir. Okay, I have seen my number and other things, sir. I typed in the answer, sir. Punganam, sir. Okay, can you check, sir? 2005. 1 minute, sir. Again, once again. Okay.
Everything coming.
Coming, sir. Okay, sir.
Yes, sir. I'm seeing, sir.
Okay, sir. Okay. Dr. Uma asked, no? Okay. 948. Yes, sir. 94860819990. Okay. Once again, thank you for all patiently here. And also kindly convey my regards to my junior, Dr. Jaiprakash. Okay, my junior and Thambi. Okay, call them all. Thank you, Pumanam sir, for the host this thing. Thank you, the participant over here.
Yes. Okay. Thank you, sir. Thank you for your wonderful presentation, sir.
Thank you, sir.
Thank you for your patience answering the questions. Thank you, sir. I will consider more of your time, sir. Thank you. Thank you, sir. Thank you,
Okay, sir. Thank you. Shall I quit now, sir?
Okay. Thank you.
Okay, sir.
I can quit, sir.
Okay, sir. Stop sharing.